everybody had a nice holiday. I've got one quick announcement for those who are listening. At the Quad AI meeting, we're going to have two pharmaceutically sponsored dinners, one on the Sunday and one on the Monday. I've sent out already the announcement for the Sunday one. I'll send it out again. And Monday, we're just getting the announcement together. I'll send that out. So if you're interested, RSVP appropriately. There'll be who and where to RSVP. And that pretty much, so the ART is on the Saturday evening. That's pretty much a full slate of events for the Quad AI meeting. Here's our speaker, so I don't have to fill in too much longer. I'll give him a minute to get himself adjusted. Anybody else have any announcements they want me to make to the general community at large? All right, well, anyway, this morning's speaker is Troy Torgerson. He's titled this, What's New in Clinical Immunology? Thank you. Right.
All right, well, thanks for this opportunity to be here. Apologize for being a little late. It's with the construction workers, it's hard to find a place to park down here. So. All right, well, what I was asked to do is to give a little bit of an update on clinical immunology, where things stand, and on some of the newer things. I'd been asked to sort of give an update on some of the screening methods that are being used for some of the immunodeficiency diseases. And so we'll talk about those. First things first, so these are my disclosures. So I direct a diagnostic lab, which we will talk a little bit about diagnostics. And I've been a consultant for Baxter Biosciences, which will play a role in one slide, which I will point out as we go on. So the goals for today's talk, I'm going to talk about three things, three updates that related to clinical immunology. So an update on screening for primary immunodeficiency diseases, PIDD, primary immunodeficiency disease. I'm going to talk about updates on genomics in primary immunodeficiency diseases, because newer genomic methods are playing a significant role in how we diagnose and care for patients, and that will increase over time. And then a little bit of an update on flow cytometry techniques and some approaches that are being used there in the diagnosis of and monitoring of primary immunodeficiency diseases. So, first things first. So this is for screening. So just as a reminder that this is sort of the breakdown of the total pie of primary immunodeficiency diseases. And the things that I'll just point out are first that antibody deficiency diseases constitute about 50% of all immunodeficiency diseases. The combined immunodeficiencies, so these are going to be things like severe combined immunodeficiency, Wiskott-Aldrich syndrome, others, about 20 to 25%, neutrophil defects 15%, and then complement deficiency, just a small group. And then, of course, there's the isolated cellular immunodeficiencies. These are largely primarily the T-cell defects, so things like the George syndrome and things like that. Okay? So this is the pie of immunodeficiency, and so when you start thinking about screening for primary immunodeficiency diseases and how you identify these patients prospectively or you aggressively identify them as you're seeing them in clinic, how do you go about doing that and having the most benefit, the most bang for your buck? Okay, and so one of the first ones, because severe combined immunodeficiency is exactly that. It's severe, and these patients are born early in and get sick early in life. And we know that it's a curable disease if we transplant the patients with SKD. There were efforts begun several years ago to come up with a method to screen for immunodeficiency diseases, and so Jennifer Puck and her colleagues were the ones who sort of had the idea initially, but the initial state that started screening newborns for severe combined immunodeficiency was Wisconsin. And and based on their experience and a lot of lobbying by families, severe combined immunodeficiency was recommended by Catherine Sebelius, the Secretary of Health, to be added to newborn screening panels as one of the diseases that is screened for in dried blood spots from babies. Okay? And that happened about 2 years ago. She recommended it. Now, the thing that you need to know about newborn screening is that this is an unfunded federal mandate. The feds basically say, we think that these diseases should be screened for. We would strongly recommend it, but we will provide the states with no funding to do any of that. So the states have to come up with the funding to screen for PKU and all those things, in addition to SKD. And so there are currently about 10 states that have gone live with newborn screening for SKD, and our state is not one of them. Just to give you the lay of the land in the Pacific Northwest, newborn screening for the state of Washington is done by our newborn screening lab. We've been working with them for about 3 years. They're very anxious to begin screening, and there is a long political process to add these diseases. And we are well into that process, and we are hoping, so it's actually been added to the governor's budget. So the Department of Health here in this state is under the executive branch. So it's been added to the governor's budget for this fiscal year. If it is passed by the legislature, we will go live with newborn screening for SKD in this state probably in July 2013 is our goal. Okay. For the rest of the Pacific Northwest, Alaska, Oregon, Idaho, all of those states, their newborn screening is done in the state screening lab of Oregon. Okay? And so Oregon State Lab screens 5 different states plus all of the provinces, I shouldn't say call them provinces, but places like Saipan, and then all of the military hospitals, and 4 of the 5 Indian Health Service hospitals are all screened in the Oregon lab. Now, we've also been in careful negotiations with the Oregon lab. They're anxious to bring newborn screening for SKD on board, and we are hoping that they also will begin screening by the middle of this year, 2013. And in bringing those 2 newborn screening labs on board, we will bring the entire Pacific Northwest on board for screening for severe combined immunodeficiency, okay, in infants. In all infants that are born in all of the states, hopefully in the Pacific Northwest. And hopefully that'll happen again by this summer. Now, how is this done? So it's this very nice DNA-based test, and so it can be done on dried blood spots. The DNA is very stable. So you'll recall that when T cells rearrange their T cell receptor as they're maturing, they go through VDJ recombination. As they do that, they loop out chunks of the chromosome, and those chunks of chromosome are joined together at these signal joint regions. So these are specific signals that signal where these loops can be cut out. And so you get, so these regions here, these signal joints, are highly conserved, okay? And what happens is that as these are looped out, it creates actually, they're ligated together as a circle of DNA. And these are called T cell receptor excision circles, okay? And these circles of DNA can be actually quantitated by using polymerase chain reaction. You use primers to these conserved regions so that you're using PCR to amplify this region that has been glued together. So one came from here, one came from here, and they've been now annealed together here to make this loop, this circle of DNA. And so you can use PCR to PCR across this junction, and it's very, very specific for these T cell receptor excision circles. Now, what you need to know is that naive T cells, they go through this process as they're rearranging their T cell receptor. They make these treks, and then the TRECs don't replicate. Okay, so as the T cells then divide, the TREC goes with one of the daughter cells and on down the line. So as you can imagine, infants have many more lymphocytes that we know than adults, about 3 times as many. So normal ALC in an infant is about 4,500. Okay, and of course infants will be largely naive, and because all those T cells are naive, virtually every T cell will have a TREC in it. Okay, so the TREC levels in an infant are very, very high because they've got lots of lymphocytes, and almost every lymphocyte has a TREC in it because it's naive. In old guys like me, my thymus is starting to involute, and my T cells have divided, and so my TREC levels are significantly lower, but they're not zero. Okay, and it turns out that in patients with SKD, where they either have no T cells, or if you've got leaky SKD, where maybe, like an Omen syndrome, where maybe just a few of the T cells are allowed to get past the developmental block and divide, those cells divide like crazy, and so they'll have no TRECs in them. And so the TREC levels in an infant will be vanishingly close to zero, if not zero, in an infant who has SKD. Sorry. So in a normal infant, the way that we quantify TRECs, TREC numbers, absolute TREC numbers based on the punch size that we make on the card, and which is a 3 millimeter punch, we'll get TREC levels in the 1,000 range, around 800 to 1,000 for an infant. For a normal adult, it's in the 300 to 600 range, and for an infant with SKD, the TREC levels will be 25 or less. So the difference between a normal infant and an infant who has SKD is enormous in this assay. And like I said, because of the way that because it detects naive T cells, it also screens for infants who might have Omen syndrome, who might have and have expanded leaky SKD with lots of expanded T cells, or they might have, or it screens for infants with SKD who may be maternally engrafted, maybe have maternal T cells that have gotten in and divided. Again, those won't have TRECs, and so it identifies all those patients. It's really, this is an amazingly robust test. Yes?
So in the oligoclonal disorders like Omen, are they halfway in their TREC production, or are they still close to zero?
So the question is, the oligoclonal disorders like Omen syndrome or leaky SKD, do they have sort of half the numbers of TRECs, or are they close to zero? And they're actually close to zero, because what happens in Omen syndrome and the leaky SKDs is that the majority of cells don't. So let's take, for instance, the RAG deficiencies, which is where the classic Omen syndrome was described. So these are where you've got RAG, the leaky patients are the ones who've got, they make the RAG protein, but it's got point mutations that make it so that it's not fully functional. It may have just a little bit of function. And so what happens is that normally, if the T and B cells aren't able to rearrange either their T cell receptor or their immunoglobulin gene, they're blocked in their development, so they can't get through. But every now and then, just by dumb luck, because you've got this sort of poorly functional enzyme, it will cut, just by chance, a T cell. And so you'll get maybe 1 or 2 or maybe a few T cells that sneak past this developmental block. And the T cells sneak past this block, and they sneak into an environment then that they come out past the block into an environment that wants T cells. And that environment has tons of interleukin-7, for instance. So IL-7 levels in a SKD patient are really super high, and so that T cell comes out into this environment and it just explosively expands. So now you've got one T cell that comes out with a TREC, but it now expands like crazy. And I think of it as sort of dropping one dandelion seed in the middle of a very fertile field. And pretty soon you've got a whole field of dandelions, but they all came from the same 1 or 2 seeds initially. And because they explosively expand, you may have normal T cell numbers, but they're oligoclonal. They're all from just a few clones, and because of that, their TRECs have been diluted. And so it's an elegant test, actually, for picking these patients out.
So I remember earlier there were IL-7 quantitative studies for screenings.
Yes.
Those didn't really pan out, did they?
No, they didn't. So the question was about the IL-7 study. So there was there was one group in California who was looking at quantitating IL-7 levels in dried blood spots, which it worked if the spots were fresh, but the protein some cytokines are not very stable. IL-7 is among those, and so as those dried blood spots would sit, the IL-7 would hydrolyze, and it would degrade over time. This being a DNA-based test, and not only a DNA-based test, but a DNA-based test that is on a circle of DNA. One of the things that makes this really robust is that nucleases that cut DNA, exonucleases or nucleases that cut from the ends of broken DNA, are much more frequent than endonucleases that can cut circles of DNA. So it makes it so this is doubly resistant to being degraded. So we can PCR DNA out of mammoths and mummies and all sorts of things. So this presumably will be very stable for years, decades even. They've gone back and looked in dried blood spots, and these are stable. So it makes this test extremely robust. Yes, Len?
Just curious, based on the number of live births in this state, how many cases you think you would diagnose in a year?
Good question. So let me back that out a little bit in terms of and answer that in a little bit of a circuitous way. So the question is, how many live births would we expect in this state per year? So the initial estimates of the incidence of SKD from Becky Buckley and Jennifer Puck and those who really sort of spent their careers studying SKD was that it was about 1 in 100,000 live births. Based on newborn screening that has been done in the states that are currently screening, it's actually closer to 1 in 35,000, 1 in 40,000. So that tells you that there were a number of kids who were born and died without ever being making it to an immunologist or a transplant center to be diagnosed. And so based on that number, 1 in 35,000, 1 in 40,000, we would estimate that in this state where the birth rate is approximately 80,000, that we would have about 2 per year. Now, what I'll tell you is that in the states that are screening, in addition to the full-blown SKD patients they're finding 1 to 2 patients for every SKD patient they find. They're finding 1 to 2 patients with significant T-cell lymphopenia. Now, some of those patients may need to go on to bone marrow transplant. Some of them may not. And that's one of the issues with newborn screening, which we really hadn't thought much about before, which is that we'd be picking up these other kids that maybe had gone undiagnosed previously, or maybe they showed up as a teenager with CVID or something. We just don't know what the natural history of some of these patients with T-cell lymphopenia was going to be. The other thing is that the first patient ever picked up with newborn screening in Wisconsin turned out to be a complete anomaly. It was a kid with an immunodeficiency who needed a bone marrow transplant, but it was the second case ever described of RAC2 deficiency, and RAC2 is actually a neutrophil defect. And why this patient was picked up on newborn screening, no one knows actually why it was the case. But the patient was picked up on newborn screening, did in fact need a bone marrow transplant, and was successfully transplanted. So we're finding out new things as we sort of screen prospectively, and it sort of raises the issue of what sorts of problems do you run into, or what sorts of opportunities do you run into screening prospectively for disease, and you end up picking up things that you didn't anticipate that you were going to pick up. Okay, so that's TREX, and that's newborn screening. Again, 10 states currently screening. The goal is to have this live across the country, hopefully in not too many years, and hopefully here in the Pacific Northwest by mid-2013. Matt?
Is this an effective test in premature births? And if so, how early?
Excellent question. So what I'll tell you is that in the Wisconsin experience, they learned pretty early on that in fact that a lot of their false positives, their false hits for SKD were in neonates who were premature, So those kids, what they ended up doing in Wisconsin is they pull those kids out and they screen them, but they don't actually report those out as SKD until they've done a secondary or tertiary screening. They actually go back and get a more mature sample when the kids are closer to their birth age or 40-week gestational age, and so that is really been the one place that this is kind of broken down, is in kids who are premature. So that's one of the caveats. Here in this state, we're what we call a 2-card state for newborn screening, which means that every child gets one at birth, and then they get a second card drawn usually at their 1 or 2 week newborn well child check with their pediatrician. So every child gets screened twice, which is not every state is like that. And so our plan here in this state is actually to screen both cards on all patients and see if that alleviates this problem with neonates. And we'll see. We know, to my knowledge, none of the states currently screening are 2-card states. Okay.
Is this an expensive test?
So doing it a newborn screening. So here in this, so I think Wisconsin added to their newborn screening panel, I think they added $5 to their panel to bring this test online. Here in this state, we've proposed, I think it's $6.50 or $7 per test to add this, but that gives us a little bit of a cushion, actually, to make sure that we've got what we need to do that. Wisconsin had a grant when they started theirs, so they didn't need to purchase equipment. They didn't need to purchase equipment and things like that off the. So we. This is a new technology which is not. These are. I didn't tell you how this is measured. It's measured by quantitative real-time PCR, and this is a new technology for newborn screening labs. They don't. Most newborn screening labs have not used this technology before, so they're having to buy new equipment to actually run this test. But doing that, as I'll show you in a few minutes, brings a whole bunch of new possibilities for newborn screening, which I'll highlight in a moment. Yes?
Is there a way that you can take the other newborn screening tests and screen them DNA instead of doing the other screens that are currently done?
So the question is whether other newborn screening tests could be screened by DNA. It's an excellent question, and I think that, yes, there are some possibilities. And in fact, some of the newborn screening labs are looking at opportunities to maybe take some of their old tests and move them in this direction, either by quantitative PCR or possibly via some of the new, more genomic type methods. And so those are things, the newborn screening labs are sort of adopting these new technologies. They've adopted mass spec in a big way. So using mass spectrometry, they are able to screen for like 50 metabolic diseases in one fell swoop off of dried blood spots. So the newborn screening labs are, as I've learned in working with them, are very, very progressive technologically and have brought on these new technologies and adopted them. And screening patients, I mean, if you think 80,000 patients in a year and you need to screen those patients twice, that's a lot of tests every day that you're running. And so the throughput in these labs is immense. And it raises whole new issues that we don't think about in a diagnostic lab like I run, where we get 600 samples a year or something. So this is this is an order of magnitude scale-wise above where we're at. So it raises some new issues. So a lot of automation. They have robotics, and they're actually really technologically advanced, these newborn screening labs. And we've got a terrific lab in this state with Mike Glass is the head of the newborn screening lab. He's got a great team, and they've been very, very aggressive at sort of developing the technology, and so they're ready to go as soon as the funding is in place. So if you have a chance to talk to your state representatives, you might just put in a pitch that as they go to Olympia this year, that maybe they support newborn screening for SKD. Because what this does is this is a chance to save lives. These kids, you transplant these kids early. Survival is nearly 100% if they're transplanted before they get infected, and you take what is a child who is absolutely going to die without question, and you turn them into a tax-paying adult, which the representatives like to hear. Okay, so that's SKD, as well as really changing a family's life. It really, I don't want to discount or be flip about the impact that this has on families. This is how the test is done. So this is quantitative PCR, and if you're familiar with it, the way that quantitative PCR works is you're amplifying the thing that you want to amplify, and each time that it's amplified, it has a fluorescent marker incorporated into that piece that's amplified. And you amplify enough until you get enough of them to sort of, that it gets bright enough in your reaction to go over a threshold limit. And so this is the threshold that's here. And so you can see here what's happened is that this is an example of using a plasmid as a control and titrating out that plasmid. So this would be, for instance, a million copies. So these are log differences in copy numbers. So this is a million copies of the plasmid, 100,000, 10,000, 1,000, and 110. And then these are where the normal patients and then the SKD patients are way down here. Because the further out you go, the fewer copies you have to start with. So that's how qPCR works. And you can make a nice plot with your known concentrations to tell where your patients lie. So this is from the initial Wisconsin work. Okay. Now, in addition to TREX, which are the T cell receptor excision circles, you can screen for CREX. And CREX are the kappa chain receptor excision circles in B cells. So this is the immunoglobulin receptor now that's being rearranged. Same story. You make a loop of DNA with these signal joints at the end. They get plugged together, and you can PCR amplify these as well. But instead of quantifying T cells, now you're quantifying B cells. Okay? And some screening labs that are quite progressive have now said, well, we're already running the test. We can actually multiplex this assay. It adds really pennies to the cost of doing this test. And so we're going to screen in every baby not only T cell deficiency, but we're going to screen for B cell deficiency. And in the pilot studies that have been done, some interesting things have come out of this. And so this is a paper from Leonard Hammerstrom's lab and showing what happens when you take patients of various types and you screen them both for CREX, so B cells, and you screen them for TREX, okay? And so again, increasing numbers of CREX this direction, increasing numbers of TREX this direction. And so here's how the patients shake out. So the red patients are SKD patients, okay? So down here you've got the patients who are T negative and B negative. So patients with RAG deficiency, okay? And then up here you've got the patients who are SKD patients who have B cells. So these are the T negative, B positive SKD patients, okay? They come out in this quadrant because they have B cells, so their CREX are positive, but their TREX are negative. So all of these SKD patients these are all T negative, B positive SKDs up here, okay? Down here, now, you have the X-linked A gamma globulinemia patients because they have T cells, but they don't have B cells, okay? And so their CREX are negative, but their TREX are positive, okay? And interestingly, one of the surprises that came out of this is that you can also prospectively screen for ataxia telangiectasia. And it's not entirely clear why, but one of the hypotheses is that because their problem is with DNA repair, the ataxia telangiectasia patients, ATM is involved in double-stranded break repair. The idea is that maybe those signal joints, they get cut out, but when they need to glue together to make the circle, they can't glue back together. And so the ends are flapping out here, and so you can't PCR amplify that little piece that you need to PCR amplify. So this is a way to prospectively screen for ataxia telangiectasia. And so you see that's here in this darker color. They're actually here because, again, if they can't glue together the CREX, they can't glue together the TREX, and those patients fall out here, okay? And then, of course, the CVID patients, some of them are abnormal as well. And so that's how this is being used. If you multiplex it and do TREX and CREX together, this is again from Swedish data. The Japanese have begun using this multiplex data, and there are pilots to add CREX screening to newborn screening in Wisconsin and in New York. They're two of the states that have been screening for a while. Because again, adding one more primer pair to a PCR reaction is pennies. If you're already running the PCR reaction, then that adds very little to the cost of the reaction. And so this is a way that you can screen for many more diseases with really minimal increase in cost. Okay, so that's CREX screening, and we'll see what happens. Now, one of the problems with screening for more diseases, cost-wise, from a newborn screening lab, is that for every patient you identify, there's a number of sort of behind-the-scenes things that have to take place in terms of follow-up, contacting the pediatrician, and there's FTE involved in doing that. And so, even though, it's easy for us to say, oh, it adds pennies to the cost of newborn screening, Well, that's true. It adds pennies to the cost of the screening reaction, but the background, the FTE involved to contact the pediatrician and the families and get them in to see an immunologist and all of that actually is not trivial. And so that's, the newborn screening labs are sort of hesitant to just say, oh yeah, it's easy, we'll just add that in, because of the cost on the back end for them. Bill?
Do the CREX and TREX have any function by themselves, I mean, in influencing the immune response?
Not that we know of. So the question is, do the CREX and TREX by themselves have any function? And we're not aware that they have any significant function. Because in an adult, they, 75% of my T cells no longer have TREX in them. And, I do well most of the time. So, anyway, yes?
I know you said the age of the T cells made a difference, but are any of these tests going to be available for amniocentesis screening?
Good question. So the question is, are any of these questions going to be available for amniocentesis screening? And, the problem with that is that these two tests really need to be done on the baby's lymphocytes. Which is a much riskier procedure prenatally, because you have to do actually do an umbilical vein tap. And so for routine amniocentesis screening, these particular tests will not. The question, though, is, at some point in the future, will we be doing broader genetic screening on amniocentesis? And I think that's very likely in the future, that a prospective genetic screening by mutation analysis will, in fact, become live in the not too distant future using sort of next generation sequencing technologies, would be my guess. Good question. So let me move on. Along the same thing of quantitative PCR, if you're going to be PCRing these two, well, why not screen for some other things? So it turns out, so again, this is data piloted and developed by the Wisconsin group, Jack Rudis and colleagues there, who did the initial screening pilot for SKD in Wisconsin. They said, well, what about DeGeorge syndrome? Because DeGeorge patients have a lot of other problems in addition to their immunodeficiency. They've got cardiac defects, sometimes neurologic defects, other things that bring them to the attention, and they need to be brought, particularly for the cardiac defects, they need to be brought to attention of care providers early. And remember that DeGeorge syndrome is 22q11 deletion syndrome, and in this region that gets deleted, there's one gene that sort of lies in the center of that region called TBX1, which is thought to be important in this process of T cell development. And so they said, well, what if we do quantitative PCR on DNA for the presence of the TBX1 gene? We'll just quantitate the presence of TBX1. So currently, the way that we screen for DeGeorge syndrome, the original way was to use FISH, so in situ hybridization on patient cells, using a FISH probe to look for whether they had two chromosomes that lit up with the FISH probe. And that's time-consuming, it's labor-intensive, and somebody's got to actually look at it under the microscope. So the next way that people were doing it was just ordering comparative genomic hybridization, so CGH array. And this is sort of where people have gone, the geneticists have gone, instead of ordering a karyotype, they do quantitative CGH array. And so normally, so here's patients, all of these patients had 2 copies of the TBX1 locus this is by a CGH array. These patients had only 1 copy, so they had a deletion of 1 allele. If they had 0 copies, they'd be down here. But these patients had only 1 copy, so these patients had DeGeorge syndrome. So this was by CGH array, but again, CGH array, it screens the whole genome. It's a little bit expensive. I don't know, it's probably about $1,000 per test to do CGH array. And so Jack Rudis and colleagues said, well, why don't we just add this into the qPCR assay, and we'll, instead of doing TREX and CREX alone, we'll do TREX, CREX, and DeGeorge syndrome all in one reaction. And so they developed a qPCR assay using amplifying for the TBX1 gene, and lo and behold, it was just as sensitive at picking up DeGeorge syndrome patients as was the comparative genomic hybridization. So again, here's patients with with 2 TBX1 alleles, here's patients with 1 TBX1 allele who have George syndrome. Okay? And here's the correlation coefficient. You see the correlation was terrific between the two assays. So now we're talking about potentially screening using quantitative PCR, this new technology, for three or more. I mean, using 3 primer sets, but to screen for more than 3 diseases. And so these are very powerful approaches. Okay, so these are sort of DNA-based techniques. Now, the guys who do proteins said, well, we don't want to be left out, and so let me show you some data that we've generated in collaboration with Si-Hoon Han. Our thought was, well, the state newborn screening labs already are using mass spectrometry to screen for, they've already got mass spectrometers, they're already using mass spec to screen for these metabolites in patients with metabolic disorders. How about if we just have them look for proteins as well? And you can do that. And so the way that you do it is that you take the protein and you chop it up with trypsin and you look for peptides. And so we said, well, let's see if we can identify immunodeficiency by looking for the absence of certain proteins. And so we set out these 3 targets. So we said for SKD, we're going to look for CD3, which is on all T cells. We're going to look for BTK, which is a marker for B cells. And we're going to look for WAS, for the WAS protein, which is obviously in all myeloid lymphoid cells, but is a marker for is absent in about 70% of patients with Wiskott-Aldrich syndrome. So in doing that, we thought we might be able to screen for SKD, XLA, and WAS in one fell swoop, using sort of a proteomics-based approach on dried blood spots, hopefully. But initially, we did it just on blood cells. And so there are all these characteristics that. So what we did is we took those proteins, we chopped them up with trypsin, and then what you have to do is you have to look for a marker peptide that serves as a reliable marker for that particular protein, and then using mass spec, you look for the presence or absence of that peptide in the chopped up dried blood spot or lysate from the lymphocytes. And so the peptides that we used for the marker had to meet all these criteria, and I won't go through those in detail. Here's the peptides that we chose. We actually chose 3 peptides for each protein. And we tested it on some known cell lines. And so these cell lines were provided. My lab put together the cell lines, fed them in a blinded fashion to Si-Hoon's lab that then trypsinized them and did the mass spec. And they were picked out the cell lines 100% accurately in terms of the cell lines that were either T cells, B cells, or were lacking WAS. And so we said, well, what about patients? And so we fed Si-Hoon's lab blinded samples from a number of SKD, WAS, and XLA patients. And for the majority of patients, they were able to pull them out. Now, the one where there were a couple of problems, one of them was in a SKD patient who was a leaky SKD patient who actually had some T cells. And so, of course, the CD3 protein was present. And so they did not accurately identify this patient. And then there were 2 patients with WAS that they said might also, they clearly lacked the WAS protein, but they also lacked the CD3 protein. It turned out that these two patients were brothers, and they're quite lymphopenic, actually, and so they also lacked T cells. So anyway, we thought maybe using a proteomic approach, we may be able to do this. So it was successful in terms of identifying the majority of patients, so we think that this holds potential. It needs to be fine-tuned a little bit, and we're doing that. We've applied for further grant funding to do that, and we think that we may be able to expand this to screen for as many as maybe 50 diseases in one fell swoop. So that's sort of the progress of proteomics. And then getting to really remember back to the pie, sort of the largest group of primary immunodeficiency disease patients were the antibody deficiency disease patients. And so the question is, how can we identify those patients? And so there have been a group of folks talking about it for a while. Baxter took the initiative to say, well, we're willing to pilot something. We will provide a grant to pilot something. There were 5 of us who they pulled together, and we thought about maybe the best way to do that. And so Baxter did an initial pilot where they said, well, we're going to target specific providers. So pulmonologists, allergist immunologists, and ENT docs primarily, because these patients, because they get recurrent sinopulmonary infections, tend to filter to those practices, so they're sort of pre-screened. And to decrease the threshold for screening these patients because of the cost of immunoglobulin screening, we're going to offer free immunoglobulin screens, quantitative immunoglobulins, to those providers if they want to use them. So they did a little pilot where they said, if you'll send your patient to the Quest lab, we'll do free immunoglobulins. There weren't a ton of patients who took the effort to go get them done, but there were enough that it provided us with a little pilot data set to show that the hit rate was actually reasonably high in this initial screen. And so that gave us impetus to move forward, but we needed something that could be done in clinics before the patients leave, because making them go to a separate lab location and other places didn't seem like it would make sense, and the screening rate would be lower. And so the idea was, can we do this off of dried blood spots again? Well, the problem is that there was no clinically approved assay for quantitating immunoglobulins off of dried blood spots. And so Baxter searched around, contracted with Quest Labs to develop a dried blood spot assay for quantitative immunoglobulins. So they elute the proteins off of the dried blood spot and they quantitate them by ELISA. They have now validated this assay, so it's now CLIA approved. And down to the age of 16. They don't have enough controls yet below 16. And now this is being offered by Baxter, so it's free quantitative immunoglobulins screening through what they're calling the ImmunoView program. As we developed this blood card, it's got a few questions on it about what the patient's major symptoms are. There's about 10 check boxes: sinusitis, pneumonias, et cetera, You just check those off, put on the patient's initials and date of birth, your information. You have your nurse in the clinic put the dried blood spots on the card. Everything is in the packet, the lancet to lance their finger, everything is there, including the alcohol swab, the Band-Aid, everything. You put the dried blood spot, you make a dried blood spot on the card, you dry it, you stick it in the provided envelope, you drop it in the mail, postage paid and everything, and within a couple of weeks you get the results back on their quantitative IgG, IgM, IgA. Okay? So it's free quantitative immunoglobulins for any patients that you may suspect have immunodeficiency. So with the idea that we want to decrease the threshold to screen for immunodeficiency, okay, among those specific providers. And so it's being piloted again in allergist immunology offices, in ENT offices, and in pulmonology offices. So if you're interested in having these in your office, contact them, let them know. Their policy or so can provide those. Now, I'm interested in this from a research standpoint because I want to know, can we screen prospectively for immunoglobulin deficiency? Because if we can screen prospectively using dried blood spots, it opens a whole new array of possibilities. Now, unfortunately, this is not going to be a newborn screen because newborns have their mother's immunoglobulin levels, but it raises the possibility of being able to do this in a broader way if the data is supportive of this. And so if you're interested, take advantage of it and see what happens. It's being funded through a grant by Baxter, and I think that it's an interesting research test to see whether or not this will actually pan out as a screening study for hypogammaglobulinemia. Yes, ma'am.
How long did you say the turnaround time is?
It's a couple of weeks, so it's not terribly long, and you get the results back there. So none of the patient information goes to Baxter. It's all kept by Quest. Baxter and the group of us who were on the committee that sort of developed this, we will get sort of global numbers. We'll see how many patients were screened and how many had low antibody levels, but we won't see anything else. And so I think that it's an interesting idea.
Could we do less than 16 if we really wanted?
I don't know that. So that's a good question, and I don't know yet. The problem is you put the patient's date of birth on it, and I'm not sure that Quest will run them if you send them in less than 16 because they're trying to, they're sort of bound by CLIA and CAP requirements. And so it won't be long before, I am in fairly regular contact with the guys at Quest. They're working on getting enough normals in the less than 16 age range. So it won't be long before they're able to do it less than that. So I don't actually know, do you guys know what will happen if somebody sends one in less than 16?
Unfortunately, right now, they will not run the test.
So I think they'll, I don't know whether you'll get a letter back saying,
You will. You'll just get a letter back.
Certainly. Slapping your hands. Yeah. Anyway, okay. So that is the status of newborn screening, or not newborn, of sort of screening for immunodeficiency diseases. This is sort of a hot area in immunodeficiencies right now. There's a lot of things going on, and so stay tuned, because with these newer technologies, things are really exploding. Okay, so questions about screening before I move on? I've got a few more minutes, and I'd love to tell you a little bit about the genomics piece. Okay, let me tell you a little bit about genomics. Okay, we'll leave the flow cytometry for another time. So genomics. So finally, 2012, the $1,000 genome. So there were two instruments introduced in 2012, two lab instruments, sequencers, that have the potential to sequence the entire genome for $1,000 or less. Okay? Now, what I will tell you to give you an idea of what these companies are targeting, these two instruments were not introduced at the genetics meetings where they have traditionally been introduced in the past. These two instruments were introduced at last year's Consumer Electronics Show. Okay? So that's what we're talking about. And this was Reuters, the report on that. Okay, so now the problem with genomics. So there's sort of a little joke in the clinic. We're like, we don't know what the defect is. Just do whole genome, whole exome sequencing. Okay. So let me tell you the problem here. So yes, you can sequence the genome for $1,000. Okay, you can get all of the data for $1,000. But this is the sort of data that you're talking about. And some of you may have seen this analogy before when I used it. So this is Leo Tolstoy's War and Peace. It's, as many of you know, it's one of the longer novels in English, at least. It's quite long. It has about 3 million letters, okay? Now, the human genome has 3 billion bases, okay? So the human genome is 1,000 copies of War and Peace. Okay, so when you send that, you send your genome away for $1,000. You get your genome, and now you get back a hard drive. So when we send whole genome sequencing, they actually send us a hard drive back with the nearly 1 terabyte of data on that patient's genome sequence. Okay, so that's how it comes back to us when we send a genome. Okay, and so this is 1,000 copies of War and Peace that you have to sort through to find the one word that is spelled incorrectly that causes the disease. Okay. Now the problem is that as you go through this, there's going to be a whole bunch of words spelled incorrectly. Some of them they'll use the UK English spelling instead of there's some of the misspellings in the genome don't make any difference. But we also know that in the through recent published studies that in the normal human genome, on average, there are 20 new loss of function mutations in genes every generation. So each person has 20 new genetic defects when they're born. Okay? And those sometimes, so the problem is, you sort through all those and you find them and you go, Jiminies, which one of those actually causes my patient's disease? Okay? So that's a problem. Now, if you just say, I'm just going to look at the exomes or the part of the genome that encodes the proteins, well, that's significantly less. It's a couple of orders of magnitude less. So now it's just 30 million letters, but that's still 10 copies of War and Peace that you have to sift through to find the one word that's spelled incorrectly. There could be a word missing. There could be a whole chapter missing, with big deletions. So anyway, this is the problem. And that led one pundit to say, yes, it's the $1,000 genome with the $1 million analysis problem. Okay? So that's the status of genomics. But it has been, despite all of this, there are better and better ways to do this piece, the data analysis piece, and it has really led to an explosion in our knowledge and understanding of primary immunodeficiency diseases, and that has allowed us to learn a lot about human immunity. And let me just give you some examples, okay? So, because we've been able to now group disorders together to tell us a lot about the function of certain parts of the human immune system. And so there are now about 200 known genetic defects that are associated with primary immunodeficiency. We estimate that there will probably be about 500 in the end, and it's sort of a gold rush right now. So everybody's trying to do whole exome, whole genome sequencing on these weird patients that they see that don't fit neatly in one of the other boxes that we've got. And there are, on average, a new gene in immunodeficiency is published every month to every 2 months. Okay, so these are expanding rapidly. Now, grouping some things together, let me just tell you about a few of these. So let's talk first about these two. So patients who've got defects where they're susceptible to encapsulated organisms like strep pneumonia. So things that we think about when patients come in with invasive strep pneumonia infections are things like complement deficiency, congenital asplenia, but also defects in these molecules that I'll show you in a second are involved in the Tolllike receptor pathway, and I'll show you those. So MyD88, IRAK4, NEMO, and ICAP-B alpha. Patients with herpes simplex encephalitis, so now also in the Toll-like receptor pathway, TLR3, UNC93B, there's also TRIF, TRAF, and I think there's one more in this pathway that I'm not thinking of off the top of my head. So here's the Toll-like receptor pathway. So this is the Toll-like receptor pathway for bacteria, and these Toll-like receptors, these are pattern recognition receptors, and they signal through MyD88 and IRAK4 to activate NF-kappa-B, and they do that through this NF-kappa-B kinase that has NEMO and the ICAP-B kinase. And here's NF-kappa-B. It's a transcription factor that's normally in the cytoplasm, and in non-activated cells, it's held in the cytoplasm by this inhibitor molecule that sits over the nuclear localization signals on the transcription factors. And when you get activation, so these are the ones where now defects have been identified in patients, MyD88, IRAK4, and NEMO, and ICAP-B alpha. And so any of these defects that result in defective Toll-like receptor responses to bacteria all are susceptible to invasive strep pneumo infections, which tells you that part of the reason that we have Toll-like receptors to respond to bacteria, they play a very important role in managing strep pneumonia in our immune system. Okay? So on the viral side, again, this is the TLRs 3, 7, 8, and 9, and these go through a little bit different pathway. Again, some of them use MyD88, but TRAF3, UNC93B, there's TRIF in here as well, and defects in this pathway have been identified, and these defects lead to herpes virus encephalitis. Almost no other problems. So it tells you that these Toll-like receptor pathways are critical for the immune system's ability to respond to herpes viruses, and primarily to prevent us from having herpes encephalitis. And what's interesting is that many of these, because these are innate molecules and these are innate pathways, it turns out that the UNC93B isn't that critical in immune cells that it be present. It turns out that this is actually critical in the neuronal response to the viral infection. So when the HSV gets into the neuron, it's the endogenous innate response of the neuron using UNC93B to make interferons to bring in the immune response that keeps normal people from getting herpes encephalitis. Okay? So this is teaching us how even non-immune cells play a role in host defenses against pathogens. Okay? And again, this has come about because of our ability to look at these unusual patients, identify new defects, but looking at patients who've got particular clinical presentations. And in this case, it was herpes virus encephalitis. Okay? So, that's one example. Let me tell you about mammalian susceptibility to mycobacteria. Now, what we know is that all of these defects now are associated with susceptibility to invasive mycobacterial infections. So the interferon receptors, IL-12 receptors, STAT1, TYK2, GATA2. So these defects largely affect the responses of the immune cells to IL-12 and interferon gamma. Now remember that in order to make a good interferon gamma response, you need to have IL-12 around, okay? And so that's why defects in both of these pathways, so the thing that unifies the susceptibility to invasive mycobacterial disease is good IL-12 signaling pathway and good interferon gamma signaling pathway, ultimately with the idea that interferon gamma is probably the most important of those. So defects in any of these molecules here can cause, and so any of the receptors, the downstream transcription factors, the kinases here, these can cause, defects in any of these can cause invasive mycobacterial disease. And a relatively recently described group of immunodeficiencies in which autoantibodies are made against interferon gamma that neutralize interferon gamma are also susceptible to invasive mycobacterial disease. And these are invasive mycobacterial disease with non-pathogenic, what we usually consider in normal individuals, non-pathogenic Mycobacterium avium invasive disease, for instance. So the last patient we had with an autoantibody was a 55-year-old Asian woman who had autoantibodies to interferon gamma, and she had a cervical MAC osteomyelitis was her invasive mycobacterial disease with Mycobacterium avium complex. I mean, a normal individual should not have that, right? And so it makes you think that there could be defects in this pathway. So again, all of these gene defects, as we've pulled them together, have told us again, what is the purpose of these pathways in the human immune system? I've got 1 minute, let me leave you with one more because it's, sorry.
In people with invasive mycobacterial disease, can you treat them with interferon?
It
Because the
depends.
drug therapy is failing so badly, is this an option?
Excellent question. So these patients who have autoantibodies, probably not a good idea because you would just be immunizing them more. So there's a pilot underway at the NIH to treat these patients with rituximab. Patients who have interferon gamma receptor defects, there's two forms. There's an autosomal dominant and an autosomal recessive form. The autosomal recessive form, they have no receptor, and so you can give them all the interferon gamma you can buy, and they will never respond. Okay, so that doesn't help those patients at all. They need a bone marrow transplant. The autosomal dominant form, they have a little bit of function, and so those patients you can treat with interferon gamma. And because IL-12 is required for upregulating interferon gamma, the patients with IL-12 pathway defects, you can treat with interferon gamma, and it will help them clear their mycobacterium and prevent them from getting infected in the future. Great question. Okay, let me tell you about one more. And it is this one, and it's chronic mucocutaneous candidiasis. So you've heard a lot about IL-17, Th17 cells, and we know about their role in autoimmunity and things like that, but the question is, what the heck is their role in normal human immunity? Well, it turns out that patients have been identified with defects in IL-17F, in the IL-17 receptor itself, in STAT1, STAT3, and I'll show you, these patients don't make Th17 cells. And then patients with Apposed syndrome who develop autoantibodies to IL-17 and IL-22. And it turns out that all of these patients, the thing that they have in common is that they get mucocutaneous candidiasis. Not invasive candidiasis, but recurrent mucocutaneous candidiasis. And it seems like in the normal human immune system, one of the major roles for Th17 cells and IL-17 is in preventing mucocutaneous candidal infections. Okay, so let me show you how this all works. So patients with Job syndrome or STAT3 deficiency, AIR1, these patients develop autoantibodies to IL-17 and IL-22. There's IL-17 receptor deficiency, IL-17 deficiency, and then these recently described gain-of-function mutations in STAT1, which are autosomal dominant and actually are, we think, more common than we might have suspected initially. And these patients have recurrent mucocutaneous candidiasis. And so here's an example. So here's a patient with Job syndrome, and these are the Th17 cells. When you activate the cells, before they don't make IL-17, but after you activate them, they make IL-17. And patients with Job syndrome or STAT3 deficiency don't make these Th17 cells that are making IL-17. And similarly, the STAT3 gain-of-function patients, they don't have Th17 cells. And so if you've got problems in making IL-17, in making Th17 cells, then you are susceptible to mucocutaneous candidiasis. Okay? And so when you get these patients in your clinic, you might think about diseases in these pathways. And the most likely one, I think, if you're thinking about these diseases, is probably, unless the patients have the other symptoms of Apposite syndrome, probably these gain-of-function mutations in STAT1, because they're autosomal dominant and they can occur sporadically out of the blue. And you only need one mutation in one allele to create a patient who's got susceptibility to mucocutaneous candidiasis and other potential problems. The phenotype is actually quite broad in those patients. So, with that, I will stop. And I will summarize by just saying that we've got a whole variety of screening tools available now. There will be more. I would encourage you to use them. They're there and available. And Genomics now are providing us the opportunity to learn about what the basic mechanisms or the basic functions or reasons for having particular pathways in our immune system are in humans, not in mice. So it's providing lots of new opportunities. Yes, Lynn.
Right, it would seem that from this new data, you may have to reevaluate your first slide. Defects in the innate immune system may be a much greater percentage than we ever recognize.
I would completely agree. Yes, I would completely agree, and it really does. You're absolutely right, because it shows really sort of, that's a slide from probably 10 years ago, and we completely ignored the innate immune system previously. And you're absolutely right that we need to go back and remake that slide without question.
Well, this was a fantastic presentation. We may have to have you twice a year.
Just to finish.
Well, thank you very much. I'll take any other questions. Thanks.
Now I have a question I don't know if everybody wants to know. I just got a lady who said,